Welcome friend to this new session on the topic of in vitro assessment of drug product performance. It is the second lecture in the lecture series that deals with the various biopharmaceutical aspect which is necessary for a drug product design. So let us start the discussion. Myself, Hassan Reza, will discuss the topic relevant to the above mentioned. We will discuss the topic which is very essential for the postgraduates, research scholars, and all the trainee researchers and research associates in various FR&Ds of pharmaceutical companies. Let us start initially with a brief historical overview of the evolution of dissolution. It was first divided into two sections. The firstly is 1897 to 1960, which actually laid the foundation of dissolution theories. Let us start with the first, that is Noyes-Whitney, which we know as Noyes-Whitney equation. It was first proposed by Noyes and Whitney in 1897 in an article called The Rate of Solution of Solid Substances in Their Own Solutions. It was first here where the rate of dissolution was made proportional to the concentration gradient. Later on, Nurst and Brown worked on the constant which involved in the Noyes-Whitney equation, specifically the diffusion coefficient, volume of dissolution fluid used, and the thickness. Later, further works were made by Wilderman in 1905, who first proposed the film theory, Hickson Crowell in 1931, who studied the release kinetics based on surface erosion. We also know about the Danckwerts surface renewal model in 1951, Higuchi, which is well known for Higuchi kinetics, was proposed in 1961. Everything which I mentioned were they elucidated the release mechanisms and dissolution rate kinetics. Later to be mentioned in this, Philbon in 1948 introduced an artificial stomach which actually stimulated the in vivo conditions, including the pH level, peristalsis, and the food in the stomach. Later on, United States Pharmacopoeia first proposed the disintegration test for an official test for the drug release in its 14th edition in 1950. So this was regarding the foundation of dissolution theories. Later on, the 1950 to 1980, it shifted towards the relationship between the dissolution and the availability of the drug in the systemic circulation, which was known as bioavailability. Edwards in 1951 suggested that the dissolution rate governs the rate of drug appearance in the body. Late in 1960s evolved the concept of the study for the extent of the drug in the systemic circulation, which was coined as bioavailability. In 1971 and also in 1968, there were two incidents that occurred that resulted in a massive change in the presumptions regarding the dissolution. It was found that in digoxin in UK and US and in the phenytoin in the case of Australia and New Zealand, it was found that there was a variation in the bioavailability and the dissolution profiles. Thus, it was suggested for an uniform dissolution parameter to be set up by officially, so that to alter the variations in the drug release which resulted in adverse reactions and toxicities. As a result of this, in 1975, 12 monographs of USP adopted officially the first dissolution tests. So this is how the dissolution evolved, and slowly it got involved or incorporated in the official monographs. First, so let us now understand the purpose of dissolution and drug release. You should remember, dissolution is a study to the rate and extent of the drug which is being released from the dosage form. The first thing which we do actually for a dissolution test is for formulation development and selection based on the in vitro drug release, which actually reflects the desired in vivo performance. It actually determines the way, the rate in which the drug has to go inside the systemic circulations. So variations are observed specifically when we are giving the drug in a controlled release, or as sustained release, or as immediate release. So it reflects the rate and extent to which the drug should reach to the systemic circulation. Secondly, batch to batch reproducibility. That's a main important thing when there is a mass production of formulation, specifically in terms of tablets or capsules. It is justified that there should be a batch to batch reproducibility of the amount of the drug in each formulation, and this can be justified by reproducibility of the dissolution rate in each batch. Next, the benchmark. It's also a benchmark for the drug product stability throughout its shelf life. So a dissolution study generally signifies that the drug is capable to release in the same release profile throughout its shelf life. So it is also a benchmark for the study for the stability of the drug. It is said that if a drug is incapable of releasing or following a definite dissolution profile, then it's not successful in releasing and showing therapeutic response. Lastly, establish a good in vitro in vivo correlation. Now, when we are saying a very good in vitro in vivo correlation, it suggests that we always try or tend to shift our dosage form to a status which has a higher solubility and higher permeability. This concept of higher solubility and higher permeability enables us to assume a bioweaver, which enables to bypass the bioequivalence studies. So in this case, a dissolution study is a better answer or a better evaluation process to whether we are able to achieve a good in vitro and in vivo correlation. Lastly, scale up for post-approval changes generally requires multi-point as well as multimedia dissolution tests, which is, you can say, is responsible for substantial biopharmaceutical implication of a product, and if necessary, bioequivalence studies are also performed to validate the dissolution test. So for scale-up process also, we need to justify the changes to be same by doing a multi-point as well as multimedia, that is using different media concentrations, in order to justify the changes if occurred during the scale-up process. Now we are coming to a point called as a dissolution test, either and how it is discriminative. Discriminative dissolution test actually is a condition which justifies the sensitivity of the dissolution method. Whether my dissolution test is sensitive to the changes which is occurring is justified by its discriminative power or capacity. So as we can say, a discriminative dissolution test is mainly done to detect or distinguish the changes in the composition and the process. Second, it's a nature that high discrimination, that if it occurs, it may result in inter-batch variations. It may cause a variation or discrimination between the batches with no changes in the viability. So this is not desired. So this is one thing which is not desirable, is high discrimination, because it may result in inter-batch variation of dissolution studies, but it will not be implied on the viability. So this is not desired. Second is a low discrimination. Low discrimination suggests that is not able to detect the changes occurring due to the formulations, but it is resulting in the changes in the in vivo process. So even this is also you can say is not desired. So what we need an optimum change or optimum balance of discrimination by using a proper dissolution test. How you can study the discriminative dissolution test? So first thing is by changing the API's type and nature, involving the particle size, area, density, polymorphs. So changing it in different way, it will affect the different drug release on the dissolution result. Next is the composition. Composition, if we suggest, so composition generally involves the way in which the drug is loaded and the nature and the type of excipients which is used. It will affect the dissolution study. Next is the process, the manufacturing process. Specifically, the type of the dosage form which you are preparing, firstly it affects, and secondly is the equipments and the machineries which are used to prepare the particular formulation. So this may also affect the dissolution test. Stability is also effective in affecting the dissolution process, as we discussed previously. So mainly if we say of stability, it is the temperature and humidity which individually or cumulatively affects the stability, and also causes an alteration in the dissolution process. So when we do if my dissolution process is discriminative, so they will be affected if any of this is changed. Now we are coming for the validation of the dissolution and the drug release. So firstly, I like to say what kind of formulation generally undergoes a dissolution test. Specifically to be mentioned, tablets, capsules, MR, that is modified release, extended release products, along with some transdermals, ointments, creams. They have some different process. So these formulations are generally are used to study the dissolution. And the dissolution process generally involves three things. Firstly is the apparatus, that is the dissolution test apparatus what we are using. Next is the medium, the dissolution fluid which is used, and the test conditions. The test conditions means the RPM, the sampling interval, and you can say the nature, even the nature and the type of the dissolution medium. So it is together known as the dissolution process. One thing we have to always remember that my dissolution process should be sufficiently rugged, reproducible from day to day, and interlab operations. That suggests that whatever the dissolution test followed by a particular analyst in one lab should be reproducible to another lab, maybe in another place, by a separate analyst. So that gives the ruggedness or reproducibility of the particular dissolution process. As per USP National Formulary, it is specified separate apparatus for specific formulations. Here I mention. So you have type 1 apparatus, that is basket type, which is used for immediate release IR, delayed release, that is DR, extended release, that is your you can say ER, paddle the same, reciprocating cylinder, it is mainly used for immediate release and extended release. Other set of apparatus like paddle over disc, cylinder, these are all used for transdermal. There are some non-compendial models like rotating bottle, which is used exclusively for extended release beads, and fans diffusion cell, which is used for studying the diffusion for the formulations like transdermals, ointments, and creams. Now, what are the factors affecting the development and validation of dissolution test? So you should remember that in a dissolution test, the validation, one of the factor is always its sensitivity and its ruggedness. So you should always remember the sensitivity that we said, like discriminating dissolution test, the discriminative nature of the dissolution test, that is one, and number two is the ruggedness. These are the two factors which are very essential. The first thing which comes to us is agitation. We should remember the agitation of low RPM. If you are using a low RPM, that is 50 to 60, specifically paddles. Now if you use a basket, it is having a lower hydrodynamics than a paddle. So obviously if you consider a paddle, a low RPM of 50 to 70 is applicable of more discrimination or produce sensitivity in the justification or finding the various formulation factors affecting the dissolution. High RPM specifically is necessary for certain formulations like suspensions comprising of viscous substances, which generally results in the settling at the bottom of your dissolution jar and producing the cone effect. So cone effect means your materials or your drugs get settled at the bottom of the paddle, producing a hill-like structure called as the cone. Now when it is producing a cone, the drug release from the cone gets reduced due to the lump formation at the bottom of your dissolution jar. So that's what for those a high RPM is required. Next is the temperature. Always for oral it is 37 degrees C has to maintain. For transdermal it is maintained to 32 degrees C. Now regarding the vessel is concerned, there is a specification for the shape and size of the vessel, and you should be highly specific with the nature of the vessel which is used. Nature and the type or shape changing in the vessel generally affects the rate and extent of dissolution. Now next is the position of the tablet in the vessel. That is very very essential. As I told you in the first point, like if the tablet is at the bottom, it is having a less effect of the hydrodynamics. So that's why there will be a relatively less dissolution. So as per the study by computational fluid dynamics, it shows that the agitation forces in the dissolution vessel, the shear force is in a small region directly below the paddle. So paddle's bottom, the paddle center is very low, as I told you, and that's why generally it is found to increase significantly if you just move at least 10 millimeter from that point. So if you just move little bit from that bottom area, if it is kept at least 10 millimeter away from that central bottom of the paddle, so it is capable of producing a better dissolution. Now for another alternative is there, but that is non-monographic, that is using a peaked bottom. There will be a peak shape, so even though you are putting the tablet inside, so automatically the tablet will not reach to the bottom of the paddle, it will be little bit away from it. So this will enable you can say to have a greater dissolution. But when you make this peak, it reduces some volume of the fluid, so that has to be substantiated. Next, for the light or floating drug delivery systems, it is very difficult that the formulation floats. Floating formulation does not produce a good dissolution. So for these cases, sinkers are used. These are the coiled metal wares inside which your formulation is placed, and it enables to sink those formulations which are floating in nature. Now you are saying of dissolution medium, of course it is the solubility of the drug in the medium. The drug should be soluble in the medium. Next, which is essential, is the medium must not get saturated in order to maintain a sink condition. So generally in a dissolution process we try to maintain a sink condition. So in no way during the drug release the medium should get saturated with the drug. So that's why a certain amount, a higher amount of medium is generally being preferred to prevent the saturation. Next, the volume of the medium should be larger enough so that it should be more, so that the volume required to dissolve the drug is used for the dissolution. The mediums which are generally used, we have deaerated water. In certain cases we use aqueous buffers of pH 4 to 8, dilute hydrochloric acid of 0.1 normal, even simulated gastric or intestinal fluid as your dissolution medium. These are some of your compendial or standard official dissolution apparatus as per IP, USP, BP and EP. So you should try to remember these names, remaining names, as type 1, type 2, type 3, type 4, type 5, type 6, type 7. Specifically these 7 types are available for USP, and for almost all, that is IP, USP, BP and EP, you can find that type 1 and type 2 is common. Next, which is also very important thing for discussion regarding dissolution process, is the calibration of the process. Two things are very essential. Firstly is the performance verification test, and secondly is the mechanical calibration, specifically for apparatus 1 and 2. Performance verification test is, you can say, is actually a test which is being performed to see the functioning of the dissolution process in that apparatus is occurring properly or not. So determining the acceptable performance of the dissolution of the test assembly, whatever you are using, And to do the performance verification test, depending upon the nature of the apparatus, you have a specific test drug which is used as a reference standard, and we see to what extent the dissolution is occurring. So if it is within the range as specified by the monograph of official monograph, then I can say that my dissolution process is calibrated. For that, we use reference standards. That is, for apparatus 1 and apparatus 2, we use prednisolone tablets, and for apparatus 3, we use chlorpheniramine maleate extended release tablet. These tablets are exclusively USP grade and should be procured from the regulatory bodies. And these are not, you should always remember, these tablets are not the marketed tablets. So, for performance verification test, specific prednisolone tablets should be procured from USP. Likewise, for apparatus 3, the chlorpheniramine maleate extended release also should be procured from USP. Now let us go for mechanical calibration. Mechanical calibration is the mechanical checking the mechanical status of the dissolution apparatus as per the requirement. There are 4 things which are generally checked. Firstly is the alignment. Alignment means whether it is erect, that is whether it is erect and whether it is at whether your shaft on vessels are aligned properly, and whether the paddle or your shaft, specifically the shaft which is used for basket and paddle, is being centrally placed. So for that we use different instruments for checking the shaft and vessel alignment. We use digital protractometer which checks whether the shaft of the vessel are at 90 degree. Secondly, for checking the centering, we use a centering gauge, and the centering gauge allows a deviation of not more than 2 millimeter. Next is the wobbling. Wobbling is a typical rotation that is being produced by basket of the paddle, in which it rotates in its axis and also makes a rotation in a circular way. That is, you can say it's a kind of dancing movement. If you see some of the apparatus, if it is not being prepared as per monograph, you will find the shaft is a little bit dancing. So this wobbling for basket of the paddle is being checked by digital test indicators, and it says that the deflection should not be more than 1 millimeter. Next is a rotation check of RPM, that is whether the RPM reading is coming correct as per the rotation of the shaft. So for that we use a specific instrument called a tachometer, and a deviation of plus minus 1 RPM is allowable. Lastly, we have temperature, for which we use a digital thermometer calibrated, and it allows a deviation of plus minus 0.4 degree Celsius. So these are some of the calibration process which is being used. Thus this ends the topic for our discussion. Hope so we are able to give you a brief overview regarding the dissolution aspects which are essential for a formulation